Good morning to all the EAPCET aspirants. So, here we are in our second session of transport in plants. In our previous session, we have dealt with both passive transport, active transport, learning complete details of diffusion, facilitated diffusion and osmosis, followed by we have also learnt about active transport. Okay, we have also learnt about active transport. And then now if we have to see the long distance transport, prior to that we have learnt to many concepts also like imbibition, root pressure, everything we have learnt. Now the second set of this chapter includes long distance transport, which includes the transport through mechanical tissues, okay, or vascular tissues. Vascular tissues like xylem and phloem, where bulk flow comes into the picture, both water and mineral transport and phloem transport, okay, which occurs in bulk. This is also addressed as bulk flow or mass flow. Once if you have to see the theory of this, diffusion is a slow process, it accounts only short distance of movement, okay. Diffusion addresses only short distance of movement. For example, movement of molecules across typical plant cell, across a typical plant cell, movement of molecules, that is known as what? Diffusion, which is short distance transport. This takes about how much time? 2.5 seconds. Long distance transport systems are necessary to move substances faster across longer distances. Fine, right? Movement of substances in this bulk from one point to another due to pressure differences between 2 points is addressed as mass flow or bulk flow. Okay, movement of the substances in bulk is addressed as bulk flow or mass flow. Like water, minerals and some food are generally moved by mass flow system or bulk flow system. Now in this bulk flow system, what happens? Substances which are transported are usually in a solution or in a suspension, right, which are of different concentration gradients. So they are swept along at the same pace like how they are in a flowing river. But in diffusion, different substances move independently, different substances will move independently depending on their concentration gradient. Bulk flow is achieved either through positive hydrostatic pressure gradient or it can also be achieved through negative hydrostatic pressure gradient. Fine, right? So there is nothing like moving across the concentration gradient or against the concentration gradient. Anyhow, the movement of substances in bulk is possible either through positive hydrostatic pressure gradient or negative hydrostatic pressure gradient. Fine, right? Bulk movement of substances in long distance through the conducting tissue. The conducting tissue is xylem and phloem. This is known as translocation. Okay, this is addressed as translocation. Clear till here? Any doubt anywhere, ma? Now, absorption of water by plants. Absorption of water and minerals, this occurs by diffusion. Okay, absorption of water and minerals occurs by diffusion. This is through millions of root hairs, okay, and at their root tips. Root hairs increase their surface area for absorption of maximum water and minerals. Okay, first root hairs will increase their surface area where they can absorb maximum water and minerals. The absorbed water is moved into the deeper root layers by two pathways. One pathway is apoplastic pathway and the second pathway is symplastic pathway. Okay, apoplastic pathway and symplastic pathway. These are two pathways through which the water is being transported across the cell walls or through cytoplasmic streaming. Clear? Then, absorption of water by plants. It is the system of adjacent cell walls that is continuous. This is continuous except at the Casparian stripes. So here from anatomy of a root system, we have clearly learned that Casparian stripes are formed at the wall layers of endodermis. These are suberin depositions. Suberin depositions. Okay, suberin depositions over endodermal walls. Both the radial and tangential wall layers of endodermis have the suberin depositions which form Casparian stripes. So, it is the system of adjacent cell walls that is continuous except at the Casparian stripes of the endodermis in the roots. Fine, right? So, what is first apoplastic movement and symplastic movement? Apoplastic movement is the movement of water across the cell wall spaces. Okay? Through the cell wall spaces, if the water is being translocated, that is the apoplastic movement. If the water is being transported through the intercellular spaces, okay, through cytoplasmic streaming, that is known as symplastic movement. Apoplastic movement is stopped at the Casparian stripes region of the endodermis. Till here, both apoplastic and symplastic movement of water and minerals is observed till the endodermal Casparian stripes. But from here, only symplastic movement is diverted. It is diverted to symplastic movement completely. Apoplastic movement is stopped. This is because of the Casparian stripes. Clear till here? Now, apoplastic pathway. Here if you see, water movement is through apoplast. This is dependent on gradient. Okay, this is dependent on gradient, and this occurs through mass flow or bulk flow. Fine, this occurs through mass flow or bulk flow. The apoplast does not provide any barrier here to water movement initially. Okay, as water evaporates into the intercellular spaces and atmosphere, tension is developed. Okay, tension develops that the continuous stream of water in the apoplast, hence mass flow of water occurs due to adhesive and cohesive forces of water. Adhesive forces is adhesion between water molecules and wall layers. Cohesion is the binding of water molecules itself. Okay, attachment of water molecules, movement of water molecules, in between them cohesion forces will happen. Adhesion between water molecules and wall layers, this gives you the adhesion force. Both cohesion and adhesion forces of water will help the water to be driven into the endodermis. Clear till here? Now, so in the system of interconnected protoplast, in this pathway water travels through the cytoplasm also through intercellular movement. This is through what? Plasmodesmatal connections. Two cells are connected to each other by what? Plasmodesmatal connections. Two cells are connected to each other by plasmodesmatal junctions or plasmodesmatal connections through which symplastic pathway is done. Water is transported through symplastic pathway there. So, water has to enter the cells through cell membrane, hence the movement is slower, movement of water is slower. Here movement is down the potential gradient. Symplastic movement may be aided by cytoplasmic streaming as well. They may be aided by cytoplasmic streaming also. For example, in hydrilla leaf, movement of chloroplast due to cytoplasmic streaming is easily visible. One of the example is in hydrilla. Next is symplastic pathway. Most of the water flow in roots occurs via apoplast, since the cortical cells are loosely packed, so water can move without any resistance. However, endodermis is impervious to water. This is due to Casparian stripes. Endodermis is impervious to water. A band of suberized matrix, so water molecules are directed to non-suberized wall regions here. Okay. The water then moves through the symplast and again crosses the membrane to reach the xylem. Okay. It again crosses the membrane to reach the xylem tissue. Fine, right. So, this is how the apoplastic movement and symplastic movement will be driven in the root system. Here if you see in few root systems where fungal associations will be there, we will call it as mycorrhiza, where root caps, root hairs will be absent. Here if you see, root caps at the tips will be absent over here, wherein Few plants like pinus, gymnosperms like pinus, they will be having fungal association. Fungal association called as mycorrhiza. Fungal association known as mycorrhiza. This is the symbiotic association of fungus with the root system. Fungal filaments, they form network around the young root and they will penetrate through the root cells. Hyphae will absorb water, mineral ions from the soil. Roots will provide sugar and nitrogenous compounds to mycorrhiza. Some plants will have obligate association with mycorrhiza. For example, pinus seeds cannot germinate and establish without mycorrhiza. Remember that. Okay. Fine. Clear till here? Next. About water movement up a plant. First concept we have to address is root pressure. Here the first concept is root pressure. Water moves up a stem against the gravity, so it needs some energy. Fine. As ions move from the soil, like just now we have learnt about apoplastic pathway and symplastic pathway of water and mineral ions. As they move across the xylem tissue, as ions from the soil actively be transported to the vascular tissues of the roots, water flows its potential gradient and increases the root pressure inside the xylem. This will increase the pressure inside the xylem. This positive pressure is known as what? Root pressure. This positive pressure is known as root pressure. It helps to push the water to some heights in the stem. Okay. This will help to push the water to some heights in the stem. Fine, right? So, the experiment to prove a root pressure, like usually during morning times, during early morning you can see the water droplets over the leaf scales, over the margins of the leaves. This is due to the phenomenon called as guttation. What is guttation? What is guttation? At night and early mornings evaporation will be low. So, excess water collects as droplets around special openings of the veins and tips of the grass blades and leaves of many herbaceous plants. Hence, water loss in liquid phase is called as what? Guttation. Water loss in liquid phase during early mornings, this is called as what? Guttation. Remember, many a time statement based questions are asked on guttation. Water movement up a plant. Root pressure can only provide modest push in the water transport, hence there is no major role in water movement. Greatest role of root pressure is to re-establish continuous chains of water molecules in xylem, which often breaks under tensions, which is created by transpiration. In most plants, majority of water transport occurs by transpiration pull. Majority of water transport is by what? Transpiration pull. Water movement up a plant. So, first is through root pressure, second is by transpiration pull. In plants, water flow upwards through the xylem. This achieves fairly at higher rates, like up to 15 meters per hour, that much higher rate is there. Water is mainly pulled through the plant, and the driving force for this is cohesion, tension, transpiration, pull again. Adhesion between water molecules and the wall layer of the xylem tissues, and cohesion, that is attachment of water molecules itself. Fine, right? Cohesion, tension, transpiration, pull model. Cohesion, tension, transpiration, pull model. Fine, right? Yeah. Next, transport, how transpiration is taking place. Okay, we know transpiration is nothing but evaporatory loss or evaporative loss from the leaves, okay, through stomata. Okay, evaporative loss of water molecules from leaves through stomata is what? Transpiration. So when we have to go through transpiration, we should be having clear idea on stomatal opening and closure. Okay, stomatal opening and closure. Usually stomata open during daytime and close during night time. This type of stomata is known as photoactive stomata. Photoactive stomata. Fine. And stomata opens or closes, opening or closing of stomata due to change in the turgidity of guard cells. This opening and closing of stomata, this is due to change in turgidity of guard cells. Inner walls of guard cell lining stomatal aperture is thick and elastic, whereas outer wall layer of the guard cell will be thin. And here the guard cells are having cellulosic microfibrils which will aid in the opening and closing of the stomata. Okay, guard cells will have what? Cellulosic microfibrils which aids in opening and closing of stomata. When turgidity of guard cells increases, the outer walls will bulge. Outer walls will bulge out and they will pull the inner walls into the crescent shape. Inner walls are pulled to the crescent shape like this, which will help in the opening of stomata. This will aid in opening of stomata. Cellulosic microfibrils in the guard cells, they are oriented radially rather than longitudinally. They are completely oriented radially like this. This will make it easier for stomata to open. The guard cells will lose the turgidity due to water loss or water stress. Inner walls will regain the original shape. As a result, the stomata will close. Usually, the lower surface of dicot leaf has greater number of stomata. Lower surface of the dicot leaf, it will be having greater number of stomata. In monocot leaf, they are about equal on both the surfaces. Okay, they are about equal of both the surfaces. Factors affecting transpiration. External factors, if you see, temperature, light, humidity and wind. These are the external factors which affect transpiration. Plant factors, number and distribution of stomata, number of stomata open and water status of the plant and canopy structures. These are the plant factors. And external factors include temperature, light, humidity and wind. Okay. Next. Here we go. The transpiration driven ascent of xylem sap. Ascent of sap, which is very important concept. Ascent of sap which is driven, water molecules driven from the root tissue to the tip of the plant. This is by three forces. One is cohesion. Cohesion is nothing but mutual attraction between water molecules. Second is the adhesion, that is attraction of water molecules to polar surfaces. Attraction of water molecules to the polar surfaces, for example, surface of the tracheary elements. Next is surface tension. In the liquid phase, water molecules are more attracted. In liquid phase, water molecules are more attracted to each other than in the gaseous phase. Clear till here? And then transpiration. The properties given, the properties give water high tensile strength, which is ability to resist a pulling force, and high capillarity also, which is the ability to rise in the thin tubes. Fine, right? So, this will be raising high tensile strength and high capillarity. In plants, capillarity is aided in the small diameter. Capillarity is aided by small diameter of tracheary elements, the tracheids and vessel elements here. Okay, this is through adhesion, cohesion, which is and tension forces, surface tension, which is increasing tensile strength and capillarity. Next is transpiration. Xylem vessels from root to leaf vein supply water for photosynthesis. This will supply water for photosynthesis. There is a continuous thin film of water over the cells. So, water evaporates through stomata where water pulls into the leaf from xylem. In atmosphere, concentration of water vapor is lower than that of substomatal cavity, fine, and intercellular spaces. This also helps water to diffuse into the surrounding air. This creates a pull. Okay, this creates a pull. The forces generated by transpiration can create pressures sufficient to lift a xylem. Okay, xylem sized column of water which is around 130 meters high. All these statements are directly from your text, which are important, right? And now, yeah, transpiration and photosynthesis, a compromise. Okay, on this you also have a short answer type of question which is very important, many a times asked in your previous year exam. So look into the theory properly. Fine. Photosynthesis is limited by available water which is swiftly depleted by transpiration. Yes or no? True, right? Photosynthesis is limited by available water which is swiftly depleted, which is decreased immediately by transpiration process. The humidity of rainforest is mainly due to the cycling of water. This is mainly due to cycling of water from root to leaf to atmosphere and then back to soil. This is a continuous cycle. So here C4 plant system helps to maximize availability of carbon dioxide and minimize the water loss. Okay. C4 photosynthetic system where a Kranz anatomy is developed in that is a good adaptation to maximize carbon dioxide availability and minimize water loss. So a C4 plant is twice efficient than C3 plant, true, right? C4 plant is twice efficient because it has Kranz anatomy in the bundle sheath cells, Kranz anatomy, and twice carbon dioxide fixation, 2 times carbon dioxide fixation occurs. 2 times carbon dioxide fixation occurs, 1 time in the mesophyll cells and second in the bundle sheath cells. Twice carbon dioxide fixation will occur. So in C4 plants it is twice as efficient as C3 plant in fixing carbon. However, it loses only half as much water as a C3 plant for same amount of carbon dioxide fixed. Fine. Next. Uses of transpiration. This creates transpiration pull for absorption and transport. This supplies water for photosynthesis, transports minerals from soil to all the parts of the plant. This cools the leaf surface, sometimes 10 to 15 degrees also by the process of evaporation. This maintains the shape and structure of the plants by keeping the cells turgid all the time. Okay, these are the main uses of transpiration. Now, uptake and transport of mineral nutrients. How this happens? Uptake of mineral nutrients. Mostly minerals are actively absorbed by roots because minerals occurs in charged particles or ions. Minerals occurs as charged particles or ions which cannot move across the cell membrane, and concentration of soil minerals is low than that of roots. Fine, right? And active uptake of ions is partly responsible for water potential gradient in the roots and therefore for the uptake of water by osmosis. Some ions are absorbed passively also. Okay? Some ions are absorbed passively. Most of the ions are driven into the root system by active transport. Next, the specific membrane proteins of root hairs actively pump ions from soil into the epidermis. So, mostly they are entering through what? Pumps, active pumps, where they require the energy ATP, that is through active transport. Endodermal cell membrane also has transport proteins. They allow some solutes to cross across the membrane. Okay, they will allow some solutes to cross the membrane but not others. These proteins are the control points where a plant adjusts quantity and types. The plant will adjust quantity and types of the solutes that reach the xylem. Next, the suberin in the root epidermis. Suberin in the root epidermis allows the active transport of ions in the one direction only. Fine, right? Suberin in the root endodermis, this will allow the active transport of ions only in one direction. Next, translocation of mineral ions. The ions reached in the xylem are further transported to all parts of the plant through the transpiration system. Chief sinks of mineral elements, growing regions such as apical and lateral meristems, young leaves, developing flowers, fruits and seeds, and storage organs. These are the chief sinks. So, remember, always the photosynthetes are translocated from source. Source will always be a mature leaf. Mature leaf. From source, they will be transported to different sinks. Okay. They will be translocated to different sinks. Chief sinks of mineral elements will be growing regions such as apical buds and lateral meristems, young leaves, developing flowers, fruits and storage organs. To these, the photosynthetes will be translocated. And in xylem, it will be unidirectional transport of water and mineral ions, but in phloem tissue, the transport or translocation of photosynthate is multidirectional. Here we go with the translocation of mineral ions only. Unloading of mineral ions occurs at a fine vein endings. They will occur at fine vein endings through diffusion and active uptake by the cells. Mineral ions are also frequently remobilized, and particularly they are from old or senescent parts to the younger parts. This is how the mineral ions will be transported. Elements most readily mobilized are phosphorus, sulfur, nitrogen and potassium. Some elements that are structural components like calcium, they are not remobilized again. Okay. Once they are deposited, it will be there itself. They are not remobilized again. Now we will come across the phloem transport. Yeah, it is a long distance transport of what? Organic substances. Okay, phloem transport is a long distance transport of organic substances like food and primarily sucrose from source to sink through the phloem. The source and sink, this may be reversed depending on the season or plant's needs. For example, in early spring, okay, in early spring, the sugar stored in the roots is moved to tree buds. Sugar stored in the roots is moved to tree buds for growth and development of photosynthetic apparatus. Thus, root becomes the source and buds will become the sink. Shoot buds or root buds. Sorry. Shoot buds or leaf buds, they will become the sink. Direction of movement in the phloem, this can be bidirectional, both upwards and downwards, because sinks will be present all across the plant, and from source it will be translocated to different sink regions. So, it can be upwards or downwards, which is bidirectional. In xylem, the movement is always upwards. Xylem, the movement of water is always upwards, which is unidirectional. Hence, food in the phloem sap can be transported in any direction, whereas phloem sap mainly water and sucrose, but other sugars, hormones and amino acids, they are also translocated over here. Mass flow or pressure flow hypothesis. It is hypothesis that explains mechanism of translocation of sugar, which is phloem transport. Mechanism of translocation of sugar, phloem transport. The glucose prepared at the source is converted to what? Sucrose, which is a disaccharide. Glucose to be stored, glucose if it has to be stored, it will be stored in the form of starch. if it has to be translocated, it will be translocated in the form of sucrose. Fine. Pressure flow or mass flow hypothesis. Here sucrose is moved to companion cells. Sucrose is moved to companion cells and then into the living tissues like living phloem. They are translocated to companion cells and living phloem sieve tubes. By what? By active transport, that is by loading. It produces a hypertonic condition in the phloem. Water potential will decreases in the phloem. Fine, right? Sieve tube cells from long tubes with holes. Here you can see sieve tube cells with long tubes with holes in sieve plates. Cytoplasmic strands will pass through these holes. They are forming a continuous filaments. Here can you see how the translocation of sucrose is happening all across the sieve tubes. Fine. Next, water in the adjacent xylem, moves into the phloem by the process of osmosis. Water from the xylem, it moves to the phloem by osmosis, or hydrostatic pressure which builds up the phloem sap will move to the areas of lower osmotic pressure. The source from the phloem sap actively moves into the cell and the cell will convert the sugar into the energy, starch or cellulose, which is a complex carbohydrate. As sugars are removed, osmotic pressure will decrease, water potential will increase and water moves out of the phloem. So, last if you see, what is pressure flow hypothesis? Identification of tissue that transport food. How the food is being transported. This is observed by one experiment called as griddling or ringing experiment. What we have to do? Remove the bark as a ring here. Can you see? Remove the bark totally, including phloem layer. After few weeks, the portion of bark will swell. Okay, this region will be swollen. This swollen tissue will symbolize that phloem is the tissue which is responsible for translocation of food, and this transport takes place in one direction towards the roots itself. Fine, right? This swollen tissue will represent that phloem is the tissue which is conducting food material or organic substances. Okay. Phloem conducts clear till here? Yeah. So, with this transport in plants is done. In our next class, we will be solving EAPCET PYQs of transport in plants, followed by mineral nutrition and the questions from mineral nutrition also. And after that, we will be looking into the photosynthesis, respiration and PGD, which ends the plant physiology chapter. Fine, right? So, that is all for today's session and thank you.